Welcome to Biochemistry of Metabolic Processes. Today, we are going to explore one of the most fascinating and vital systems in all of biology, the mitochondrial respiratory chain. This is the microscopic engine inside your cells that keeps you alive, thinking, and moving. Let us dive in and see how it works. To start, imagine you are watching a highly coordinated relay race, but instead of runners passing a baton, it is a team of specialized workers passing a high-energy package. In our bodies, this package is an electron, and the workers are tiny molecular machines. As each worker catches and passes the electron to the next, they use a little bit of the electron's energy to do some heavy lifting, like pumping water up a hill. At the very end of the line, the final worker hands the exhausted electron to oxygen, which safely tucks it away by making a drop of water. Now that we have a mental picture of this relay race, let us look at the actual biological structures involved. The mitochondrial respiratory chain is located within the inner membrane of the mitochondrion. It consists of a series of 4 large protein complexes and 2 mobile electron carriers embedded right into this membrane. These components are perfectly positioned to work together. They execute a sequence of oxidation reduction, or redox reactions. They accept high-energy electrons from molecules like NADH and FADH2, which were generated during earlier stages of metabolism, such as the citric acid cycle. With the components in place, let us understand exactly what they do with those electrons. As electrons flow through complex 1, complex 3, and complex 4, they release energy. The complexes actively use this released energy to pump protons, or H+ ions, from the mitochondrial matrix into the intermembrane space. Think of this just like pumping water up a hill. By moving protons against their concentration, the cell creates a powerful electrochemical gradient across the inner mitochondrial membrane. This gradient is a form of stored potential energy waiting to be unleashed. This stored energy gradient brings us to the ultimate purpose of the respiratory chain. The cell uses this powerful electrochemical gradient to synthesize ATP, which is the primary energy currency of the cell. Without this microscopic electron relay, our bodies would be completely unable to extract enough energy from the food we eat to sustain complex life. While processes like glycolysis produce a tiny fraction of our energy, the respiratory chain is responsible for generating over 90% of the ATP in a healthy human body. It is the true powerhouse of our existence. To truly appreciate how critical this continuous flow of electrons is, consider what happens when it stops. A stark real-world example is cyanide poisoning. Cyanide is deadly because it binds directly to the final protein complex in this respiratory chain. By doing so, it instantly blocks the final transfer of electrons to oxygen. The entire relay race comes to a crashing halt. Because the electrons cannot flow, no protons are pumped, the gradient collapses, and ATP production stops completely. Without ATP, cellular functions fail rapidly, leading to rapid death. Now that we understand the biological mechanism, let us look at the thermodynamics that drive it. Electrons flow because of a difference in reduction potential between the donor and the acceptor. We calculate this driving force using a simple subtraction. The formula is delta E0 prime equals E0 prime of the acceptor minus E0 prime of the donor. Delta E0 prime is the total change in standard reduction potential, measured in volts. A larger positive difference means there is a stronger driving force pulling the electrons from the donor to the acceptor. Let us apply this formula to a concrete example, the oxidation of NADH. We are given that the standard reduction potential for the NAD plus to NADH half reaction is negative 0.32 volts. For the oxygen to water half reaction, it is positive 0.82 volts. We set up our calculation by placing the value for oxygen, the final acceptor, first. Then we subtract the value for NADH, the initial donor. Subtracting a negative number effectively adds the magnitudes together. This gives us a highly positive potential difference of 1.14 volts, indicating a very strong driving force toward oxygen. With the potential difference calculated, we can now determine exactly how much energy is released to pump protons. We use the relationship between standard free energy change and reduction potential. The formula is delta G naught prime equals negative N times F times delta E naught prime. Delta G naught prime is the usable energy released, measured in joules per mole. The variable N is the number of electrons being transferred. F is the Faraday constant, which is approximately 96,485 joules per volt per mole. As the potential difference increases, the free energy change becomes more negative, meaning more energy is released. Let us continue our NADH example and calculate the total energy released. We know 2 electrons are passed from NADH to oxygen, so N equals 2. We plug in the Faraday constant and our previously calculated potential difference of 1.14 volts. Multiplying these together gives us approximately negative 220,000 joules per mole, or negative 220 kilojoules per mole. The negative sign confirms this is an exergonic reaction that releases energy. This massive release of energy is enough to drive the synthesis of roughly 2.5 molecules of ATP per molecule of NADH. Now, let us see what happens when electrons enter the chain slightly later, via FADH2 at complex 2. The donor potential for FADH2 is 0.05 volts. Subtracting this from oxygen's potential gives a smaller difference of 0.77 volts. We plug this new potential difference into our free energy equation, again using 2 electrons. The result is approximately negative 149 kilojoules per mole. Because FADH2 provides less energy than NADH, fewer protons are pumped, ultimately yielding only about 1.5 molecules of ATP per molecule of FADH2. Having calculated the energy, let us visualize the entire system in action. Here you are looking at a schematic of the inner mitochondrial membrane. Notice the 4 large protein complexes embedded in the gray membrane layer. Follow the arrows representing the electrons. They enter at complex 1 from NADH or complex 2 from FADH2 and are shuttled by mobile carriers, Q and cytochrome C. As the electrons travel horizontally to the right, look at the vertical arrows pointing upward. These represent the protons being pumped from the matrix below into the intermembrane space above. Finally, at complex 4 on the far right, the electrons meet oxygen to safely form water. We have covered a lot of ground, so let us summarize the most important ideas. The mitochondrial respiratory chain is a vital pathway that safely extracts energy from electrons donated by the nutrients we eat. By passing these electrons through specialized complexes, the cell captures energy in manageable steps to pump protons across a membrane. This creates a powerful gradient that sets the stage for massive ATP production, providing over 90% of our body's energy. Understanding this chain reveals exactly how oxygen keeps us alive and how our bodies harness the hidden energy within our food. You now have a solid understanding of this incredible biological engine.